Hi, my name is Avi Ma'ayan. I'm the PI of the Mount Sinai KMC. This is an update of what we've been up to in the past year for the face-to-face meeting. So for this meeting, we prepared 6 posters, each describing a tool that was developed by the lab. And here I'm going to just give you a brief overview of those projects. So when you search for gene on Google, you get a lot of links to different websites that potentially provide information about those specific genes. But it's pretty hard to go through each of those websites and figure out what is there and what you might be missing. It's nice to see that Harmonisome is showing up as the first entry for search for this particular GPR 173. So how do you know whether you are going to be missing potentially informative information about a gene landing page from a Google search? So for this, we developed this new website called Gene and Drug Landing Page Aggregator, where we assembled landing pages for genes from 50 sources. And also we have 19 drug repositories. And you can come here and you put in information about gene or drug, and then you receive the results as cards that give you links to the landing pages from those databases. So for example, here I clicked on a GPR 173, and if I click on the Gene Mania page, I directly get this network for GPR 173 that provide information about genes that are co-expressed or physically interact with this particular GPR. And this is the poster that will be presented by Max. So in the past, we show you how you can get high quality predictions for genes based on gene co-expression. And one of those landing pages is the ARCHES-4 gene landing pages that we have developed. And those landing pages have been highly successful. They've been accessed over 3 million times so far. So this is the page for GPR 173, predicting the goal biological processes that suggest neuronal activity and specifically vesicle-related neuronal functions. Last year, we showed how we can improve gene function predictions by partitioning the gene-gene co-expression network into clusters and forming gene-gene co-expression network on each cluster. And this is sort of like mimic the concept of different cell types. Co-expression is changing in different cellular contexts. By combining that information, we are improving the quality of those predictions. And then the more clusters we make all the way up to like 300 clusters, we see improvement in those predictions. But how can we make those predictions relevant to biologists? So typically when you do differential expression analysis, you perform gene set enrichment analysis. And here you can see how we perform gene set enrichment analysis on our bulk RNA-seq samples from biopsies from patients with diabetes and comparing those to patients with diabetes and HIV. And we get enrichment for kidney development. So the idea is now to bring in with bridgeGSEA the dark kinases, ion channels, and GPCRs into the fold of the analysis. And those are shown in magenta lines and also in the bar chart shown on the right. And we can see that both predicted to be a part of kidney development based on co-expression, but are also highly ranked in the context of this particular study. So to learn more about this method, Alexander Lachman is going to describe this bridgeGSEA method that he developed. So the predictions by ARCHES-4 and PRISM-X are done to coding genes. So we cover all the human coding genes, but we recently applied this approach to also non-coding genes. So for this, we developed this website called LinkHub II, where it serves knowledge and predictions about over 15,000 long non-coding RNAs. And those pages are presented as reports. You get the secondary structure of the long non-coding RNA, the co-expressed genes, the co-expressed other long non-coding RNAs, as well as biological functions, pathways, and mammalian phenotype predictions based on the co-expression approach. The report also provides expression across tissues and cell lines, as well as visualization of the long non-coding RNA in the space of all other long non-coding RNAs, as well as predictions for drugs that potentially would increase or decrease the expression of the target long non-coding RNA. In this particular case, I'm showing the results for hot air, which is a well-studied long non-coding RNA. And those predictions are made by the Link's L1000 data. So to learn more about this poster, Megan Wojciechowicz is going to describe the work that she's doing for mining knowledge about long non-coding RNAs, which are mostly understudied. So to create those reports, Megan developed Appyters, and those Appyters are a way to rapidly convert Jupyter notebooks into web-based bioinformatics applications. Two of those Appyters allow you to enter a gene, and then it automatically identifies the different perturbations that maximally increase or decrease the expression of that gene. So the first Appyter was created by mining gene expression from GEO. So to do that, we first counted the tokens of labels for each sample from all of the GSMs of human and mouse studies. So in general, the labeling of samples on GEO is very messy. So you can see that some of those short terms, for example, control, is likely representing the control samples, DMSO, wild type, while other, for example, treated or knockout, those are going to label the samples that are the treated or the conditioned samples. So we can't cover all of the samples, we can't label all of them, but for those samples that it's easy to label, we were able to automatically label them and using regular expression, extract the controlled perturbation of those from those studies and generate over 8,000 gene expression signatures that are all served on this Appyter, where you come in with your gene, for example, GPR 173, and then you can find studies from GEO that maximally up or down regulate the expression of this gene. For example, here you can see that the drug Aleve, which is a pain NSAID, significantly down-regulating the expression of GPR 173. The second Appyter is using the Link's L1000 data in a very similar way. We created over 1 million gene expression signatures for small molecules and genetic perturbations from the Link's L1000 data. And for example, for GPR 173, we can see that there are CRISPR knockouts followed by expression that downregulate the expression of GPR 173, and one of them is another GPR, GPR-C6A, which is a known GPR that is a part of the androgen system. So this poster will be presented by Sherry Jie, and she will describe you the construction of those two Appyters and how you can use them. So the connectivity map approach for links. The typical idea behind it is that you come in with your up and down differentially expressed genes, and then you can identify small molecules that can potentially reverse or mimic your input signature. And recently, like I said, we processed all of the newest LINKS L1000 data to create signatures from it. And here you can come in with your up and down differentially expressed genes, the results of the top mimickers and reversers. For example, here, what I've done is I queried GPR 173, but the genes that are mostly co-expressed with it. And then here are the ranked drugs that are mostly reverse or mimic the expression of those co-expressed genes. And one of those drugs, mitoxantrone, is consistently showing as reverser of the signature of the genes that are co-expressed with GPR 173. So the website that hosts all of those signatures, this new search engine that we've developed from the links data, is called SigCom Links, and Errol Evangelista will be presenting a poster that describes this new resource. So the last project I'm going to describe is called SigCam Graph, and it's a continuation of the SigCam Links project. With SigCam Graph, you can create workflows starting from a gene set, a disease, a drug, and by selecting, for example, a gene, I can build a workflow that expand knowledge about that genes for multiple sources. For example, I can get a figure from Arches 4 about the expression of GPR 173 across tissues. I can expand GPR 173 with additional genes based on the literature or co-expression. From those genes, I can select the top 50 genes to create a gene set. And then I can analyze that gene set, for example, with tools such as GSEA to identify transcription factors that regulate those genes. Or I can analyze those genes with enricher. And again, those are genes that co-express with GPR 173. Finally, I can export this workflow as a report. And this report gives me visualization of all of the results that I produced, including the GSEA analysis and the enrichment analysis all in one place. So this is an automated way to create very rapidly workflows to form hypotheses about gene sets, diseases, and drugs, drawing from APIs that are served across many tools and databases that we and others developed. The poster for this project is presented by Daniel Clark, who is actively developing this system. I also would like to mention several new publications. One of those papers is a collaborative work with the other KMC. We created 14 tutorials about how to use the different resources that have been developed by the KMC over the years. We also published a paper about DrugShot, who you probably heard about before. It is a version of GeneShot, but for drugs. So basically you come in here and you type in any search term, any PubMed search term, and then the PubMed search term is turned into a ranked list of drugs based on co-mentions. And also you can get predictions about additional drugs based on co-expression. And finally, a paper that was just published in Journal Bioinformatics presents fast implementation of the GSEA method. And this is critical when you're doing thousands of GSEA tests. So lastly, I would like to acknowledge all the members of the lab and the funding from the NIH.